keep in mind that it's not a real thorough QT study in the sense that you don't have a positive control. So you don't necessarily administer moxifloxacin as part of this first-in-human trials. You don't necessarily have also the same power that you would require in the normal thorough QT. And obviously we also need to keep in mind that if you don't see any QT prolongation as part of the first-in-human trial, that might not be sufficient to actually have a waiver to conduct one thorough QT study later on during your development. But it's clear that this might constitute some good supportive data, especially for those molecules for which you may actually be able to convince the agency that a thorough QT might not be required later on. So one thing I think that will become maybe more prevalent in the future. I just provided here some examples of frequent questions or questions that we were used to see during the last couple of years from the ethics committee. So question about the justification of time allowed between cohorts, something that I've mentioned before. So again, if you are very aggressive and have, for example, a 1-week interval between each cohort, it's very important, again, to have a good rationale to support that in terms of safety evaluation, safety committee meeting, and what are the provisions to allow you to go ahead and dose a cohort very quickly after the first one, for example. Justification of the time allowed between the sentinel or the sentinel cohorts and the remaining subjects of the cohort. So, for example, if the plan is to dose on day 1 2 subjects and on day 2 the remaining cohort, the IRB had questioned why we were not a little bit extending this period of time between the two to allow a better safety evaluation. Justification for not using a staggered dosing approach. Justification in the case of an integrated protocol for starting the MAD portion of the study before the SAD portion was completed. So again, it's very important to provide all proper justifications in terms of safety in order to avoid questions from the ethics committee. And the dose escalation criteria, especially in the case where you would be somewhat aggressive in terms of dose escalation scheme. Very important, again, to support that with some safety consideration in the protocol. In terms of reviews, since our clinical sites are based in Canada, I've included some questions I had from the clinical team of Health Canada, and it's quite similar to some of the questions I've shown you in the previous slide from the IRB. But we've seen other types of questions. So clarification, justification for the upper age limit. That's the example I've talked to you about earlier during my presentation, where there was a recommendation to increase the upper age limit. Clarification regarding the selection of the starting dose. Again, dose escalation scheme, stopping criteria, the stopping rules in the protocol. Allowing more time between each cohort. Example I've mentioned before, where the agency actually asked that we use 14 days instead of 7 days between each cohort. And also each subject within the same day, as also mentioned by one of the participants today. And that's about the questions from Health Canada. I focused my talk on true first-in-human studies, but obviously many people consider phase 1 trials as almost any trials that are conducted in healthy volunteers, and not necessarily trials that are conducted only at the beginning of clinical development, because some of those clinical pharmacology studies might be conducted during phase 2 or phase 3, or even sometimes after the submission to the health authorities. But this is just a variety, obviously, of different types of clinical pharmacology studies that are often conducted, in most of them in healthy volunteers. It's interesting to note that even though for first-in-human trials there's not so many guidance documents, and I think there's probably good reason for that, besides the one on dose escalation, on dose calculation from the FDA and the EMEA guidance on first-in-human, there's not so many guidance with regards to the design of first-in-human. But with regards to clinical pharmacology trial, there are many guidance out there that help sponsors and CROs to design those studies. So